Uh, in the last 2 days, we completed DNA concept, DNA RNA concepts of gene, genetic engineering, and today we'll start and complete cloning and the transgenic plant. Is that clear?
Yes.
Okay. Next. For tomorrow, genome and gene library. Okay, cloning. Next. Okay. Now, what do you mean by cloning? The word cloning is an art or is a process, and the cloning is derived from the word clone, and the meaning of the clone is a dical.
Identical.
Aim.
Same.
Similar.
Similar.
Is that clear? The meaning of the clone is always going to be similar or same or identical. Is that clear?
Yes.
Is that clear? So they are similar, and that's why they are clone of each other. What?
Clone of each other.
Next. Okay. So clone. The meaning of the clone is having the similar copies. Now, what do you understand by clone is a similar? Now, how you get this similarity? What was the similar about those two people? They were Jasba was same. Is that clear? They were same externally. Their phenotypic character is same. Phenotypic character is nothing but the external character. When the external character is going to be same, then why this happen? Why the two people or more than two people having the same external character? They are having the same external character because unka internal jasba is same. That is they have a same genotype. When the two organisms going to have a same genotype, they are always going to have a same phenotype because it is genotype which control the
Phenotype.
Is that clear?
Yes.
Okay. So we can have identical gene, that is called as a gene cloning. We can have identical cell, that is called cell cloning. We can have identical organism, that is a complete Vijay is going to be different Vijay. Is that clear? Two cellmen can. Two similar cellmen can. Is that clear? Is that clear? Is that clear? So this is called as they have a same genotype. The whole body, the whole animal, the whole organism remains the same, and that's why it is called animal cloning or the whole organism cloning. So clone may be gene cloning, clone may be cell cloning, clone may be organism cloning or the whole organism cloning. Is that clear? Next. You can always create a clone of unicellular organism, of multicellular organism, of prokaryotic, of eukaryotic, of plant, of animal, anybody. But the meaning of the clone, the word clone always apply to the living organism or the living thing. If you have identical chairs, you cannot say the word they are clone of each other because the word clone is always applied to the
Living things.
living ing,
Living things.
living king.
Living things.
Is that clear? Is that clear? Now I hope you understand unicellular organisms are made up of single cell, multicellular organism made up of many cell. Prokaryotic does not contain nucleus, but nuclear material is present. Eukaryotic contain true nucleus, that is nuclear membrane is present. Plant, animal, you understand all the things. Is that clear?
Yes.
Next. So this, if you do not understand, this is prokaryotic cell, this is eukaryotic cell. In prokaryotic cell, nuclear membrane की लगी
What?
न्यूक्लियर मेम्ब्रेन की लगी
What?
डीएनए की हुई चांदनी रात में साइटोप्लाज्म के साथ छैया छैया करते हुए तो फिर क्या बात दही के साथ रात मिलेगा दाल भात. Is that clear? But in case of eukaryotic cell is a true cell. U is a true, karyon is a nucleus. Having such a or true nucleus is eukaryotic. समझता है? मतलब they contain nuclear membrane and everything. All the structures are going to be present in the nucleus. So you can always create a clone or identical for the prokaryotic, for eukaryotic, for the animal which are made up of single cell, amoeba, paramecium, multicellular, that is we are multicellular. You can create the whole organism cloning that is exactly identical to me. We can create animal. Is that clear? Next. Now we are going to focus on to the gene cloning and the microbial cloning. Now focus on to the gene cloning. I already explained you genetic engineering is nothing but the gene cloning. Is that clear in the last lecture? Clear? Now what is gene cloning? Gene is a gene. Cloning is the process of making identical. The process of making identical in
Gene.
the process of making identical gene is called as a gene cloning. Is that clear? Now I already explained you the first step of genetic engineering is the isolation of gene. Second step is always going to be the selection of the vector, then insertion into the vector. Is that clear? Then this is called as a recombinant plasmid. Is that clear? Recombinant plasmid. Super cool. Now this recombinant plasmid is sent into the bacteria. That is a host cell. This bacteria, B for Taria and B for? Is that clear? So B for Borivali as well. So this is nothing but the Borivali. Now once this recombinant plasmid enter inside, what I explain you, I need to multiply this particular gene because this gene is a human gene. Which gene? Insulin gene. Is that clear? Is that clear? So what exactly happened? I already explained you. This particular plasmid is a self-replicating plasmid. Self-replicating matlab 1 to 2, 2 to 4, 4 to 8. तो मतलब वो खुद से डिवाइड हो सकते हैं. संगीता समझता है? Is that clear? Is that clear? Now वो खुद से डिवाइड नहीं भी होते हैं. The whole organism is going to multiply. That is called, this is called microbial cloning. When the one plasmid containing human insulin will divide, then the whole insulin gene will be divided. Is that clear? This is called as a gene cloning. That is, genes are getting multiplied. Cloning is the process of making identical. And when we are making the identical of gene, this is called gene cloning. Clear? Is that clear? Now what is microbial cloning? In the microbial cloning, the whole microorganism become to multiply. This is only a one microorganism. And this is the only one microorganism cannot give me the large amount of insulin. There are millions of people who are dying because of insulin, Sangeeta. Millions of people dying because of diabetes mellitus. There are millions of people who are dying because of deficiency of insulin, Sangeeta. Is that clear? Is that clear? So we need to multiply this microorganism, and when we want to multiply this microorganism, we need to provide them the food and the nutrition. Is that clear? So when this particular bacteria is provided with the food and nutrient, लगातार this bacteria from 1 to 2, 2 to 4 and 4 to 8 लगातार संगीता वो मल्टीप्लाई हो जाएंगे और सारी दुनिया को इंसुलिन मिलेगा और इस दुनिया में किसी को डायबिटीज नहीं हो पाएगा. संगीता समझता है क्या? समझता है? नेक्स्ट.